We're now moving to a 5-minute virtual flash talk from Dr. John Eleblu, who's going to be talking about cowpeas. So I wonder whether we can have that 5-minute presentation now, please.
Good day. My name is John Eleblu. I am a senior lecturer at the Biotechnology Centre and also coordinator for research programs Francophone Africa at the West Africa Centre for Crop Improvement we call WACCI. Cowpea is an important multi-purpose legume crop in Ghana. It is one of the priority crops under the Planting for Food and Jobs program. The main objective is to develop platforms for tissue culture-based genetic improvement of cowpea in Africa at WACCI. This is to enhance research capacity and institutional capacity for research. And so WACCI, NIAB, and Cambridge Africa are the main partners. This is the picture of Dr. Alison Bentley sharing a joke with Her Excellency Royal Majesty Queen of England, Queen Elizabeth, and just before showing her the cowpea plants that were regenerated in vitro, and you can see some of the cowpea plants there in the glass jars. This occurred during the centenary celebrations in 2019 when the Queen of England visited the laboratory of NIAB. Results from the work done so far at NIAB, we've been able to set up the platforms in collaboration with partners to have regeneration of whole cowpea plants from explants taken from the cotyledons. And so successful and reproducible regeneration has been achieved from cotyledonary explants excised from 4-day-old seedlings. As you see, shoot initiation, and then at day 3, you have elongation after 2 weeks and induction of roots after 6 days. Plants are acclimatized in growth chambers, and then you have seed to seed achieved in only 3 months in controlled environment. You see the protocol has been optimized at NIAB to support the regeneration of whole plants from explants. So this allows the whole regeneration of cowpea plants, and so we are excited about this. And at WACCI, we've been able to optimize the protocols and produce cowpea plants in vitro. After germinating them in vitro, we develop the explants and then regenerate whole plants on media that we can even supplement with organic additives. Instead of using hormones, we use tomato juice, coconut juice, and other organic additives, and we're successful in regenerating plants using cheaper alternatives. So the protocol has been optimized and Ghanaian accessions have been tested. We found the most amenable to tissue culture, and we are now ready for the plant transformation step of the project. And so we call for support and for funding to help us develop cowpea materials or varieties with extreme drought resistance. This is in hope of developing materials that can be more resilient in our growing conditions in the arid regions, because desert plants have the ability to survive extreme droughts. They hold their processes until there is water, and then they regenerate new leaves and then bear and produce. We hope these resurrection genes that have been found in desert plants can be moved into cowpea to make it more resilient and then increase the yield of cowpea for better livelihoods of our smallholder farmers. And so this project is open for collaborations, it's open for more investments, and we call on all of you to support the work we are doing.